Dear colleagues, my name is Ekaterina Mironova. I am PhD, head of the Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Physiopulmonology, and senior researcher of Laboratory of Molecular Mechanism of Aging, St. Petersburg Institute of Bioregulation and Gerontology. Today we will discuss the topic how peptides regulate directed cell differentiation. In a multicellular organism, all cells are adapted to perform certain functions. Nerve cells, blood cells, bone cells, epithelial cells, and gland cells. They are all completely different. At the same time, the set of genes of all these cells is the same, with rare exception. For example, human erythrocytes do not have a nucleus, and genetic alteration occurred in immune cells. Not all genes work in each cell, but only a certain set of them. Depending on which set of genes is turned on in a particular cell, or other words to express in a given cell, it belongs to a particular cell type. All cells of a multicellular organism appear as a result of the division of one cell, a zygote. As a multicellular organism is formed, cell differentiation occurs. The switching on or off of certain genes and the specialization of the cell to perform certain functions, acquiring the characteristics of a particular cell type. As differentiation proceeds, the cell's ability to divide decreases. Most often, cells that have not acquired the specific features of a certain cell type divide in the body. An exception is, for example, hepatocytes, liver cells, which are capable of dividing in a differentiated state. This is associated with the high ability to regenerate characteristics of the liver. In a multicellular organism, tissue homeostasis is maintained by stem cells, which are undifferentiated precursors of one or more cell types. It is known that one of the manifestations of aging is a weakening of the differentiation capacity of the stem cells. Research has shown that peptides can epigenetically regulate the expression of various groups of genes, including those responsible for cell differentiation in the body. From the polypeptide complex taxarest, which was isolated from the bronchial mucosa of young animals, the tetrapeptide IADL was synthesized, which is amino acid sequence is alanine, glutamate, aspartate, lysine. The effect of IADL peptide on differentiation of human embryonic cell lung was studied using quantitative polymerase chain reaction. On the left, you can see the control group. On the right side, you see how IADL peptide change the ability of embryonic lung cells. It was found that IADL peptide promotes increased expression of genes involved in differentiation of bronchial epithelium. From the polypeptide complex suprefort, which is isolated from the pancreas of young animals, the tetrapeptide KDW was synthesized, which is amino acid sequence is lysine, glutamate, aspartate, tryptophan. It has been established that with the aging of pancreatic cells, the expression of differentiation markers in them decreases. On the left side, you see the control group. On the right side, you see a group of pancreatic cell culture with KW peptide addition. Tetrapeptide KW stimulates the expression of differentiation factors of acinar and islet pancreatic cells in young and old cultures. From the polypeptide complex Vladanix, which was isolated from the thymus gland of young animals, the dipeptide Ke was synthesized, which is amino acid sequence is lysine glutamate. The effect of Vladanix on the differentiation of human hematopoietic stem cells was studied. On the left side, you see the control group of cells. On the right side, you see the Vladanix addition to the human hematopoietic stem cells. It was found that Vladanix reduced the expression of hematopoietic stem cells differentiation markers such as CD44 and CD117. Vladanix also increased the expression of the mature T lymphocyte marker CD28. This indicates that under the influence of Vladanix, CD117 plus cells differentiate into mature CD28 plus T lymphocytes. The effect of the dipeptide Ke on the stimulation of immune cell differentiation was investigated. On the left side, you see the microphoto of the control group, and on the right side, you can see the Ke peptide addition to the embryonic thymus cell culture. Dipeptide Ke stimulates differentiation of thymic T cell precursors toward CD4 plus cells, T helpers, in rat and human thymus cell cultures. Cell differentiation is accompanied by appearance of their ability to perform specialized functions in the body. Polypeptide complexes and short peptides included in their composition provide the maintenance of the functional properties of various body systems. The effect of the ideal peptide on lung cell differentiation may underlie the bronchoprotective effect of tax arrest in models of inflammatory diseases, fibrosis, and toxic lung damage. In turn, the differentiation of islet and acinar cells of the pancreas induced by the KEDW peptide may be one of the mechanisms in the treatment of diabetes and the anti-inflammatory effect of Supreforte in treatment of pancreatitis. In addition, the ability of Ke and Vladanix peptide to stimulate the differentiation of native lymphocyte confirms their immunomodulatory activity. Vladanix can be considered as one of the medicines for the prevention of viral infection, the effect of which is expressed in the compensatory stimulation of T lymphocytes with a decrease in immunity. Thank you for your attention.